Hi all, and thank you for joining me today. My name is Trevor Long, and I'm a graduate student at the University of North Dakota pursuing a PhD in biology. I'm excited to share with you my work in improving our understanding of the genomic mechanisms behind cell fate choices. Now, understanding cell fate choice is a complex problem because it is a multi-scale process. Gene expression is determined by interactions between multiple genes at the genomic scale, multiple enhancers at the scale of the genetic locus, and multiple binding sites at the scale of the enhancer. This process, or cis-regulatory architecture, is programmed within the cells but remains unknown to us. My ambition is to decode the cis-regulatory architecture behind cell fate choice. To start investigating this problem, we used the hematopoietic system, or more specifically, the macrophage-neutrophil differentiation, where progenitor cells differentiate into macrophages and neutrophils as a model. This differentiation is thought to be driven by the ratio-dependent interaction of PU1 and CBP alpha proteins. Increased expression of CBP alpha is a key regulator in neutrophil development, and it has been found that CBP alpha knockout mice exhibit neutropenia. Because of its importance in neutrophil development, we have been working on decoding the cis-regulatory architecture of CBP alpha during the macrophage-neutrophil differentiation. In doing so, us and others have identified several enhancers and potential binding TFs that drive the expression of CBP alpha, with enhancers 7, 16, and 18 being of particular interest. To simulate macrophage-neutrophil differentiation, we utilize a model cell line known as PUER cells in which bilineage progenitors differentiate into macrophages or neutrophils upon cytokine and tamoxifen treatments. To understand how CBP alpha expression is regulated during this differentiation, we measured the activity of the promoter and known enhancers by utilizing luciferase reporters integrated in a site-specific manner into the ROSA 26 locus using CRISPR. The CBP alpha promoter and enhancers, serum 7, 16, and 18, were individually integrated into a luciferase construct, and the resulting expression of luciferase was measured at multiple time points during differentiation. Here we see the relative expression of endogenous CBP alpha in panel 1 and the relative expression of luciferase in panels 2 through 4 at different time points throughout differentiation. In summary, we found that the activity measured in serum 7 and 18 mirrors that of the expression of endogenous CBP alpha during neutrophil development, suggesting that they are key expression regulators. To uncover the driving mechanisms behind these temporal activity patterns, we also profiled the accessibility of the CBP alpha locus and enhancers with deep sequencing at several time points during differentiation. The accessibility of regulatory regions is regarded as a determinant of gene expression since it could influence the occupancy of TF binding sites. Here we see the chromatin accessibility patterns of the 110kb CBP alpha locus with undifferentiated PUER cells in pink, macrophages in green, and neutrophils in blue. The accessibility of CBP alpha showed that no novel accessible regions appear as a result of differentiation, nor were any previous accessible regions lost. Instead, the accessible regions showed a quantitative change in accessibility, where they become more or less accessible during the different stages of differentiation, but always remain accessible to some degree. Of the accessible regions in the chromatin structure, the CBP alpha promoter was the most accessible, followed by CRM 18, 7, and finally 16. We also saw a high correlation in CRM accessibility structure between replicates and samples, which suggests that when accessible, enhancers are protected at the same binding sites throughout the differentiation. The high correlation of the enhancers and promoter allowed us to pool the samples for a very high resolution image of the regulatory regions, equating to approximately 50x coverage of the accessible murine genome, whereas typical ATAC-seq libraries have a coverage of only 0.4x for mapping accessibility to 3x for footprinting. However, it's worth noting that footprinting with a coverage of 3x has been met with mixed success in identifying TF footprints at an individual level. Because of this, many footprinting techniques have moved to the whole genome aggregate level of analysis. With 50x coverage, we saw that the accessibility pattern of the enhancers consists of many short, high accessibility islands, 1 to 5 base pairs in length, interspersed between 5 to 50 base pair long regions having nearly zero accessibility, with the CBP alpha binding site protection as an excellent example. To determine if these high-low-high patterns are indicative of individual TF binding sites, we mapped previously known binding sites and chip-seq signals to each of the enhancers. Here we found that all of the binding sites overlap a footprint and fall at or near the maximum of their respective chip-seq signal. Together, this is strong evidence that supports our ability to identify individual footprints through measured accessibility. Having determined the TF binding architecture of the CBP alpha regulatory regions, we then aimed to analyze our temporal accessibility dynamics that can describe their activity patterns. We looked at the change in total accessibility, or the sum TN5 cuts, within the promoter and enhancers during differentiation. Interestingly, we saw an increase in activity that precedes the increase in total accessibility in both CRM7 and 18. Also, we observed an increase in accessibility after OHT treatment hour 0, but a decrease in activity in CRM7 and 18. This suggests that the activity of the enhancers cannot be explained solely by their total accessibility. Therefore, we began to analyze the accessibility dynamics of each individual nucleotide or position within the enhancers. This showed that only a small subset of positions increase early with initial GCSF treatment, which are then followed by a late increase in the majority of positions with OHT induction. In CRM7, we saw that the early increases in accessibility occurred at the islands bordering CBP family binding sites, while late increases in accessibility occurred at islands bordering GFI1 binding site. CRM18 showed similar dynamics. It is thought that the height of the accessible islands surrounding TF footprints are representative of higher TF occupancy. With this, it can be inferred that the activity spike seen in both CRM7 and 18 during early neutrophil development is driven by the early binding of CBP family TFs, which is stimulated by GCSF treatment. In conclusion, our high coverage accessibility maps reveal a complex regulatory architecture with 5 to 6 TFs binding up to 20 sites in a single CBP alpha enhancer. Furthermore, the temporal dynamics of accessibility and gene expression do not support a causal role for total accessibility and gene expression. More generally, these results show that profiling gene expression and accessibility at high temporal and genomic resolution have the potential to reveal the causal drivers of gene expression changes during development. And with that, I would like to thank everyone who has helped me make all of this research possible. Thank you.